Wisconsin Lutheran College's 2013 iGem team has been hard at work to create a secretion mechanism that is safe and customizable. Our way of approaching this is to make use of purification and secretion tags that are separated by a single enzyme cut site, NHE1. This is the cut site that is the insertion point for any coding sequence of interest. Then, the induced gene can be secreted from the cytoplasm to the extracellular space via a tripartite SEC1 pump. In order to control a TSC2 toxin, a SIMAR-regulated T5-cumate promoter is placed within the plasmid. This will prevent horizontal gene transfer. Essentially, if the plasmid is ejected from the bacteria and picked up by another, the gene producing the toxin will not be suppressed and will kill the cell. The plasmid also encodes a chimeric OMP-A monoclonal antibody gene, which when expressed inserts into the outer membrane, and then the antibody then selectively binds norovirus within the gut to hinder infection. This has great implications in the way of helping those with viruses in developing nations. This plasmid construct was used to overexpress and secrete plant-based polymer-degrading enzymes BGLS, YesZ, and Zyne. This will allow us to break down hemicellulose, cellulose, and pectin. These enzymes were isolated from the cellulosome of Bacillus subtilis subtilis 168 to digest plant material. We utilize Escherichia coli Nissl 1917, containing a chromosome-integrated SIMAR gene, to secrete those enzymes in the hope to accentuate dietary uptake for humans and livestock in impoverished countries. Implications: one of the great potential benefits of this project is to provide impoverished people with more access to food. This project will allow a greater array of options for food sources, as well as increase the nutritional value of the food that is already present. The mechanism that this project provides will be extremely helpful to other iGem teams. This system can be used to secrete any other enzyme or molecule chosen by an iGem team, or even general researchers.
Reception. The intent of this project is to create a product that would aid developing countries without necessarily changing their cultures or westernizing them. In these countries, nutrition is a major concern. The lack of nutrition in developing countries has the added adverse consequence of increasing chronic diseases. Nutrition is something easily changeable that has the power to prevent chronic disease, with scientific evidence increasingly supporting the view that alterations in diet have strong effects, both positive and negative, on health throughout life. Nutrition is the first step to overall health, and therefore, our product could improve the general health of a developing country. Some sources even venture to say that food production is considered an evolutionary advancement. Culture is not static, but dynamic. It is the aim of this project to enhance the cultures that it touches. Countries also have varying concerns about GMOs, especially the UK and India. Countries like the United States, Canada, and China would be more likely to accept these products. This product would go through further testing to ensure that it is safe for consumption in order to send to countries that could highly benefit from this product. Future. Polysaccharides are a convenient energy storage system for many organisms. In order to use the energy, the organism must break down the polysaccharide chain into smaller mono and disaccharides. Cellulose is one such polysaccharide, an energy storage form for plants. Glucose monomers link together in long, rigid chains to form cellulose. The plasmid breaks down plant products that are not cellulose, but similar in structure, such as hemicellulose and pectin. We have three enzymes to accomplish this: Zyne, BGLS, and YesZ. The endoxylenase Zyne cleaves xylin polysaccharide chains to form shorter xylin chains. The enzyme YesZ has beta-galactosidase activity, cleaving endo-beta-D-galactans to produce single galactose molecules to form RG1 pectin. The endo-beta-glucanase BGLS will cleave internal 1,4 linkages adjacent to 1,3 linkages. A secretion system has been designed to release enzymes from the bacteria. In future experiments, other enzyme-producing genes will be added to break down plant material further. Cellulase genes are first in line, as cellulose is the largest composition of cell wall. The 2008 Edinburgh team designed the genes required to break down cellulose into glucose. These are exocellulase, an endocellulase, and a beta-glucosidase. Other enzymes required to fully degrade plant material would be added as well. Overall, this product and the secretion system that it provides will be advantageous and beneficial to future iGem teams as well as developing countries.